Okay, good afternoon, everybody. So good morning, good evening to everybody who's listening in virtual. Thank you for those who are here physically, who are joining us for the Health and Biomedical track for Supercomputing Asia 2022. All right. So welcome to this track. I think there is timeliness in having this track because we are an exciting age where there's an era of big data in genomics and precision medicine. So you can see that there's the ability for us to measure multiple biological parameters is really increasing. We can measure things at a population level, at the individual level. We can measure things for the single individual down to the single molecules such as metabolites. So you can see all these omics here is just opening a door for large complex data sets. An analysis of these data sets actually hopefully will lead to interesting insights for improving patient outcomes. So this is actually driving the need for computational and storage resources. And this track is hopefully to introduce you to some of the opportunities in which various researchers are harnessing HPC and AI to accelerate research and development in this area. So I'm truly pleased to be able to introduce you to three eminent speakers on these topics. So the first one, just quickly introduce all three of them, talking on real-time virus surveillance at pandemic scale. So we are pleased that Peter Bockner, who's a founder and president of GISAID, will give some introductory remarks, followed by a talk by Dr. Sebastian Maurer-Stroh, who's executive director at the Bioinformatics Institute at A*STAR. We'll follow up with also looking at the perspective of precision medicine, measuring interesting biological parameters, particularly DNA methylation at the population level. So we'll follow up with a talk by Prof. John Chambers from NTU. You can see that he wears many hats, Professor of Cardiovascular Epidemiology at the Lee Kong Chian School of Medicine at NTU. Importantly, also, he's the Chief Scientific Officer of PRECISE, which is a new office coordinating the national precision medicine efforts. And lastly, but not least, we are pleased to have Professor Daniel Ting from Sing Health. You can see he wears many hats also. He's been a key leader in actually trying to translate AI and helping to benefit patients. So with that, I'll introduce our first speaker. Okay, great. So as I mentioned before, our first speaker will be introduced by Peter Bockner, who's the president of GISAID Initiative, talking about real-time virus surveillance at pandemic scale. A talk will be delivered also by Dr. Sebastian Maurer-Stroh. I'm clear? I'm clear. Okay.
And the organizer for the opportunity to express GISAID's deep gratitude to the researchers and the government of Singapore. In particular, a very profound thanks goes out to our colleagues at the Bioinformatics Institute at A*STAR. Together you have and you continue to play one of the most critical roles in making GISAID's performance possible. The world's truly indebted to you for your remarkable contribution during this crisis that is not over yet. It was actually, I have to see, I have my screen just changed something here. Let me just move something here. Hold on. Sebastian did the full screen thing and took over my computer. Hold on. I cannot see my screen. Oh, all right. It was actually here, very much in this room that I'm in right now, in the afternoon of January 9th, 2020, where I stand before you, where this journey began for us. We had just received from our friends at the China CDC the very first whole genome sequences that GISAID got to share with the world moments later. Their data basically enabled the development of the first vaccines and the first diagnostic tests to detect SARS-CoV-2 truly in record time when you think about it. And now, a little more than 2 years later, the 9th millionth SARS-CoV-2 data set is about to be made available on GISAID. 9 million. This is an unprecedented sharing of data by 210 nations and territories, and certainly we believe a testament to the importance of GISAID's data sharing mechanism. February, which is still here today, the 28th, marks actually the third time in a row where we had over 1 million records added in a single month. So these are new dimensions. And that happened not long after the Omicron was first detected and then rapidly shared by GISAID. I don't need to tell you the enormous demand this places on reliable computing power, nor how important it has become to harness high-performance computing and AI. We need to improve the surveillance and permit real-time analysis and the evolution of the pandemic virus, as Ken alluded to moments ago. Just let me say that GISAID looks very much forward for a long and very innovative partnership with NSCC. We not only share the enthusiasm, but also the vision of Ting for the need and benefit of supercomputing in Singapore. So allow me to wish all of you successful proceedings during this important conference. And I will hand over to my close colleague, Sebastian, to share with you more details what real virus data sharing has enabled in this pandemic. I thank you for letting me greet you from California. Thank you so much. Greetings to Singapore.
Thank you very much, Peter. Thank you also again to the organizers for inviting us to contribute to the discussion. And as Ken said, it is very timely because we are still in the middle of pandemic. The virus changes, and I will talk a little bit about that, but the scale hasn't really slowed down. I'm gonna start by giving you some list of things what this real-time virus data sharing enabled, and then also the reason of how it was becoming possible. There is, of course, a big technical component to this, but there are also many other factors. That is about the trust that people have in data sharing mechanisms, which also goes beyond virus sharing, but maybe can also find its application in clinical data. So as already mentioned, as soon as the Chinese CDC submitted genome sequences that were shared via GISAID on January 10th, several different groups had access to the genomic sequence. Nowadays, technology has come so far that you only need to know the sequence so that you can start working on the vaccine. You can start ordering, synthesizing constructs to make your vaccine and start testing it out. And within record time, not just from the sharing, but then everything until approval, indeed, the vaccines became available. It is, of course, also clear that first diagnostics devices depend on knowing about the sequences. And it's not just a single sequence that is needed here, but it's a high quality set of sequences that are characteristic for the outbreak so that you know what are the characteristic sequence features that you need to detect. And as was mentioned, GISAID is benefiting from this global cooperation. GISAID is enabling this because many countries are hesitant to share data in a way that they cannot control what is happening with their contribution. And just to give you this example here, you have the classical model of full public domain databases, where you would have, you deposit your data and everyone can do with it whatever they want, including making patents, making papers out of it. Instead, the GISAID mechanism is different. It helps protect your rights to it, but you can still share it. So it's very important that you can still have public health efforts powered by this whole approach. And if you see in the upper left corner, you see that already very early on, the number of genomes that came into this data sharing platform clearly outperformed other databases. And from this rise onwards, it was going all the way here. We do believe that it's a matter of a few days until we reach the 9 million mark. And also you can see the coverage in the globe. Now, if you compare this to 2009 in the swine flu pandemic, it was also a global pandemic, but a much smaller number of countries that could submit sequences. There are now 4,200 institutions that entrust their data to GISAID and about 50,000 participating researchers, and this number is, of course, growing. GISAID is also the primary source for influenza, which was its origins, but also recently for RSV data. Combining just the SARS-CoV-2 data makes it the biggest virus sequence database in the world, outperforming all other viruses together. So HIV, influenza, RSV, And all the other viruses are less genomes available than for the SARS-CoV-2 virus. So the world really has responded with a very big effort to this. As I had mentioned, the reason behind this is the mechanism that GISAID has, which is based on granting the users a license for the use of the data. The access to GISAID is free of charge. Anyone can register, and it's open to everyone, but they need to identify themselves. So the way GISAID can enforce that the data is not being misused is by having everyone who wants to see the data to register with their own personal account. And then the only requirement for them to use the data is that they need to acknowledge the contribution of the data providers, as well as try to establish collaboration. So this is really a mechanism to foster collaboration and prevent things like scooping with manuscripts, especially for high-profile new virus data. This is something that would hinder the public health response if people hold back the data. And because of this mechanism, the virus genome sharing has become extremely fast. And that is why we are talking about real-time data sharing nowadays. For example, in old times, if we would submit something to GenMang, it could take 3 months for it to become available. And imagine if you get to see the first Omicron sequence 3 months after. So you're already overrun by the virus by that time. Instead, what during this pandemic, through the GISAID platform has been established is that rapid sharing is possible. Every country that is colored here on this map has submitted data in the last 6 months. So these are 114 countries and territories. And what is so remarkable is that they have submitted within 1 month of the sample collection date. So that means in an actionable time frame that you can understand the change and the arrival of new variants. And there are also 31 of them that do it within 2 weeks. Singapore, for example, is one of the countries that regularly submits within 1 week. And even some other countries and bigger countries like the UK are having a remarkable effort with good speed. So I had already mentioned that having this real-time data flowing through allows us to track and identify new variants. And this is the general picture just over the last year. You can see the virus, how it has changed. In the red color, this was the alpha variant. And alpha was quickly replaced by delta. And we thought this was a very quick replacement, you see here, until Omicron came with this color here. And you see the replacement has been even faster. And when we show these different colors, the differences between those are tiny changes in the genome that manifest themselves as amino acid changes also on the spike protein. And this is what drives the evolution of this difference and the emergence of new variants. You can see here, this is one of the surface proteins of the virus, so that the antibodies and also the host receptor would interact here. And antibodies from vaccine or natural infection would also recognize some other part. And then there is a cleavage site where additional molecules can attack to change the stability and the speed of the infection of the virus of the cells. So it's important that we have computational steps that translate the nucleic acid code into the proteins and into the phenotype that it makes. So we know the difference. The delta causes some different severity, infection rate, as well as Omicron. You see the number of mutations that are both delta and Omicron are away from alpha and the original Wuhan virus. And Omicron clearly seems to be winning here with a whole lot of additional mutations. Alpha, delta, Omicron are, of course, the big variants that you know, but the level of detail in the constant work and surveillance is finding the new variant, tracking the changes within the variants. And these are some of the lineages within Omicron. You've also heard about BA.2. It is very important to not always be concerned or worry that there is a new variant, because this is just the natural viral evolution. We always see new lineages. We have seen this over the last 2 years, going up and down, up and down. And but what is important is that we are aware of these changes and what they mean. And I will give two examples about that. So that is the portion where we need to continuously monitor and maybe get more genomes. You can also see that the difference between these lineages, again, are different changes here in the structure. The green ones are those that are shared among the Omicron lineages, whereas the BA2 has the purple ones here, and whereas the B1 and 1.1 has the blue mutations here. There is also a BA1.1 that is just like the BA.1, but it has one additional orange mutation here, which is where antibodies bind. So this is just some descriptive picture of that the virus constantly changes, and it doesn't necessarily mean it becomes more dangerous, but it's important to monitor and that we can respond to it. And it's important to have sufficient data with it. We are living in the time where big data and AI are powerful tools that can help us. And it's also very important that for data, just like we have here for the virus, we have a lot of metadata available from the simple systematic naming of the virus in a structured way, so that you can already, as a researcher, understand, oh, this is a virus from Israel from this year, to adding additional information, for example, full detail where it came from, the collection date, some outbreak detail. Then there are some epidemiological data that can help you making better analysis about the sampling strategy. And then, of course, there is clinical data to the level that is permitted by privacy laws in different countries. In some cases, not all of the data can be fully added if there is risk of identifying individuals. But then there are also technical information that are important, and that is important also for us computationally, because it's not just getting the sequencing done, it's the bioinformatics pipeline, how to get it further into the consensus sequence that can vary in different parts of the world, as well as in different groups. So whether it's Illumina or nanopore or even Sanger sequencing, and then what are the bioinformatics pipelines, what are amplification protocols, all this can be recorded. And then we can detect, and it has happened indeed, that we can see biases that come not from the natural evolution of the virus, but from using specific protocols. And understanding that allows us to also make them correct for this. So not fall into the risk of having a bias, but rather correct it from understanding the data. There is also the benefit of adding clinical trial information. For example, the monoclonal antibody developments or the vaccine developments of some companies. The vaccine breakthrough cases that they find in their trials are important because you can see then, through comparison with the tools that I will describe later, where are the changes that make the virus work less for certain vaccines. And this is the important part where the computational aspect comes into play. So we have a plethora of tools. Some of them are also developed by our team here in Singapore, but most of them are spread throughout the world. There are some classical tools that have been around for ages, and we have now a new challenge. We have this large number of genomes, so that we hardly can get all of this data organized and analyzed in a timely fashion if it were not for high performance computing and improved algorithms. What you see here is the analysis tools that are available for GISAID users after you log in. And I will just show some of them. And I will highlight those, of course, that are more related to the supercomputing aspect. In the field, there was a commonly used software environment that had nice graphics, but it was limited to about 4,000 genomes. This was fine when this was the amount of viruses we would see in surveillance pipelines, but it didn't take long for this to become obsolete. And bioinformatics tools at that time were not able or capable at all to do trees of this amount of genomes that we see now. So what has happened is that we and colleagues have worked on improving these pipelines, and also the developers of these tools have improved, so that now with this new virus situation, it is possible to make analysis of 6 million genomes. And this pushes forward the limits for this kind of analysis, not just for viruses, but also for other tools. When I say that to make this tree of 6 million, and these are the high quality genomes that you can see, there is multiple steps to reach. Apart from the whole pipeline of sharing the data and the database behind, once the data is there, it needs to be aligned. It needs to be brought in a relative register so that it can be compared. This alignment procedure requires high performance computing. We've been doing this for several months on our servers in BI, but this is a job that has become so big that we also need supercomputing and high performance computing to help us with this. And as a service to the community, because not everyone has this in their backyard, these alignments are being offered to, for download for the registered users. So it's also environmentally friendly if not everyone has to do the same kind of analysis, the first step of the analysis by themselves. Many of them are not able to anymore because of the scale of the problem, but this is a good function that GISAID provides to its users. And then the next step is for the tree. That is another time-intensive step that creates, allows you to compare the spread of the virus in different areas. And very much related to what do you do with this comparison? If you know what is the nearest neighbor of your genome, what can you learn from it? And here is the example where many of you will know the classical tool BLAST that has been around for decades and is an extremely simple tool that just has the job. You put in your sequence and it finds you the nearest neighbor of those sequences by sequence similarity. However, with a database of 8.6 million genomes, this search now would take over an hour. And this is not something that is necessary or needed for you to wait here. So instead, what we did together with colleagues in New Zealand was develop a new tool called Audacity Instant that does the whole job. You put in your query sequence and it makes the calculation in a few seconds. And there are a few tricks how you can achieve that, especially through proper indexing. You need to know that among the 30,000 bases, there are only few that are characteristic. So you can use tricks like suffix trees. You can use tricks like having loaded in memory the indices. And then instead of an hour, it just takes a few seconds. And this would be one example. So I show here an example where a Singapore surveillance sequence is compared against the whole database. And below is the summary of where the hits come from. So you see the majority of the nearest neighbors of this genome come from the Philippines. You also see the time window where they come from is some time before April. So it's interesting because this sequence in Singapore was collected on 15th of April, submitted on 26th of April, but the nearest neighbor from the Philippines was collected in March, but submitted a month later than the Singapore sequence. So what this shows is that a real-time fast surveillance can pick up cases or can give you an impression of what is the virus variant scenario in other countries by doing this kind of surveillance. And this surveillance here case came from when you enter Singapore and you do a test, there is also some surveillance of travel history. And because of this travel history, we can also confirm that this indeed was a traveler from the Philippines with this virus. And it shows how, especially in the beginning of the outbreak, when few cases were around, contact tracing is very important, whereas in later parts of the pandemic, it becomes more important to get the overall picture right. And these tools to go from the sequence to actionable insights, this is how and why the real-time surveillance is important and works. And the next tool I want to mention is a tool that allows you to check if your diagnostics still work. As mentioned, the very first time you see the sequence, you can start designing, make diagnostic kits. But you also need to know if the virus didn't change so that that same probe can still detect the virus. And this example that I show here was one of the earliest primers by Christian Drosten in the Charité, the polymerase primer that worked for a long time in the pandemic. But when Delta came, there was one mutation in the 3' region, which reduced the efficacy in detecting that. And the global surveillance system plus bioinformatics analysis has picked this up very early and could help colleagues adjust their prepared primers, etc. Currently, the global surveillance system for influenza and for coronavirus by WHO uses a set of primer, this US CDC SC2, that triages if a sample is part of influenza or part of coronavirus. And we have it here in our system, and we can answer questions like, we know the BA.2 sublineage in Omicron has been rising. Has it been not detected by our assays? And there was the difference already that the S gene dropout doesn't work if you know this detail or so. But what you can see is also that at the level of the individual probes, there can be a few mutations, but it's also important to see that it's just a very marginal part. So this is always good news that when you see 1 or 2 mutations, but it's not happening in the million of sequence that are there. That means your system is able to detect, but the primers are still working. So the ability to go from the sequence to what does it mean for the public health response is an important aspect. And because of that, we publish twice weekly, besides our other updates on the variants, like this graphs that I showed before, also the performance of some commonly used diagnostic kits. And going to the last component, this is also related to monitoring if your assays or countermeasures are still working. So we're talking now here about effects of these mutations on vaccines. This is something how we started working with GISAID over a decade ago. Working on a tool called the flu server allowed us to very quickly tell if there is a drug resistance mutation from the genome by doing structural modeling of it. We built a very similar tool, the Prof server for the coronavirus. It was already in its first version available in January. And essentially what it does all of the pipeline steps needed where you need expert knowledge or computational knowledge to compare the sequence, to link it with the database, to show it in the structure and do calculations. It does it for the user. And we can tell, for example, if a mutation happens at the host receptor binding site, which can affect transmissibility, or also if it happens at the antibody interface. If it happens at the antibody interface, this is how we know that a variant could potentially affect also vaccines. And if we combine this information in the 3D structure and the potential phenotype with the global surveillance system to know that a variant is rising in some parts of the world, then you can come up with an emerging variant system. And this is a system that was live on GISAID already in the months that the alpha variant came up. It was a response to seeing this variant come up. And we have trackers that as soon as a new variant comes up, we see it early. We look at the composition of the mutations, the functional relevance, and we closely monitor if they come up and if they see a potential rise in some areas. Sometimes it happens very, very fast, like Omicron was swiping through. So it's not easy always to predict well in advance, but it's definitely a way of monitoring it. It was also listed in the MIT Technology Review for COVID variant tracking as one of the top 10 breakthrough technologies in 2022. This brings me to the end of this presentation. And GISAID is possible because of the GISAID community and the trust in the data sharing system, but, and especially in this context, I need to say also a lot of computers behind. So thank you very much for giving us the opportunity to speak. And I don't know if there will be any questions. I'm happy to take them.
All right. Thank you, Sebastian and Peter. Really, this was a very interesting talk and definitely quite timely, given like this is talking about the situation that has affected us all for the past 2 years, and really like getting to know sort of like the insights that like high throughput computing, data infrastructure for data sharing, and real-time analysis can bring is really insightful. So I'd like to like remind the attendants that like you can ask a question via Pigeonhole. I think there will be a QR code flashed next to me. And so yeah. And so maybe I'll just start off with like a couple questions I had when I saw your talk. So I remember that like the Omicron variant sort of came as a surprise with a lot of variations suddenly piling up. In Insights, has there been sort of like through the GSAID efforts some sort of a variant that like did not sort of had the success of the Omicron variant, but like that popped up in places?
It's a good question. So as I mentioned in the beginning, new variants always pop up, and most of them are not successful. However, at the level of the number of changes that we've seen with Omicron, we rarely see such sequences. If we see this, usually it would be isolated and often of lower quality. Part of the GSAID mechanism is that we help the submitters improve the data, and you can also upload raw read data, and we can help making sure that it gets the best possible quality out. So in numbers, the Omicron variant had 45 mutations in addition. This in a phylogenetic tree makes it a far-off branch indeed. And normally we would see a new undetected variant just maybe 5 to 10 mutations maximally away. That was why the Omicron variant looked a bit different initially. But the important aspect is that it was detected and reported by our South African colleagues, Tulio, also with some consequences, unfortunately, about the rapid sharing. But because they shared it, a lot of additional response was possible.
Nice. Oh, so there is a gap in our understanding of like how Omicron came to emerge. It like suddenly there was this so successful variant with lots of mutation, but it's like a far-off branch of the phylogenetic tree.
That's not the topic of our of our presentation, but of course there have been already some studies on that. One of the hypotheses could be that it emerged in an immunocompromised person, or even in an alternative host like rodents. And it just needs to be undetected for a while. If it's long-lasting within one host, in a single person, it could change like we saw.
Nice. All right. One question from Stefano is: are there any planned application of GSAID to a wider comp biofield, such as protein prediction and parallel sequence alignment?
Yeah, I think a lot of the technology can be cross-seeding into other fields. In fact, the flu server that was giving rise to the cough server, we also did for the National Precision Medicine program here to look at variants that are happening near drugs in 3D protein space. And I think it's something that is very useful. And in terms of if the high performance computing components will find their way into, let's say, human genomics, I think some of the technology, but one needs to look in detail yet what can be used. But I'm always sure this technology advances in one area will push the other two.
Actually, a related question is as like progress in AI tool, especially like AlphaFold and like protein prediction structure, impacted the analysis that could be done in the context of GSAID and in the hunt for
Yeah, it's a good question. So AI analysis in general, of course, through many different angles, because you have so much clinical data, metadata associated. But AlphaFold in particular, it may not have revolutionized the response to this virus, because all of the major proteins were already known, and through homology modeling already in January 2020, we had an idea about the 3D structure. But AlphaFold is a very interesting and important tool that will continue to be important for any other outbreak. So if the next outbreak would happen, the tool I would use to make the prediction of the 3D structure would probably be AlphaFold, just in the case that it's not close enough to a known template where mostly AlphaFold will get the right answer. And I think it's always important to look at multiple tools, including the Rosetta Fold, etc., to get a consensus of the structures. So I'm positive that it will impact more pandemic response in the future for sure, these AI-based structure prediction tools.
And actually, related to that, so like GSAT is mostly focused on like sequencing of the actual virus variants, yeah? But one aspect of the virus is the differential response across people. I mean, some people get terribly affected, some much less, and also like different variants have different tropism, yeah? Omicron is much more severe because it attacks like the upper part of the respiratory tract rather than down in the lung. Are there efforts to, one, collect the genotyping of the spike protein interacting variant, and two, actually to play with like AlphaFold and those type of tools to understand what changes the tropism between different variants, and or even how some people react much more dramatically than some other to infection?
So I think this is a naturally part of the research that is being done. I think a lot of the difference that you see in people responding to the virus is not because of the virus itself, but it's because of their different immunological genetic makeup, different history of exposure to these viruses. And the Singapore NCID has published multiple studies where we were linking the genotype of the virus also to different levels of viral load, etc. But at the same time, they also studied in how severity depended on different immune response that was not triggered by the virus. So the teams of Lisa Ng from ID Labs in ASTA and also David Lai at NCID have done a lot of work on the host response. And there's also new technologies like single cell response that colleague Chian from GIS has been working on. So I think it's not happening on a global scale, but on a country scale, I know that many people are also looking at the host response. And this is something that GISAID tries to support by adding the clinical trial and patient data information as much as the privacy laws permit.
I see. All right. Well, thanks again, Sebastian, and thanks, Peter, for this interesting talk. I'll basically leave it to Kenneth to introduce the next speaker.
All right. Thanks a lot, Sebastian, and also Peter, for introducing, GSAID, the role of GSAID, and the potential applications of HPC and AI moving forward. Next, we are pleased to introduce Professor John Chambers. He'll be talking on deconvoluting genetic regulation of DNA methylation. So as I mentioned before, Prof. John Chambers wears many hats in Lee Kong Chian School of Medicine. He's also the Chief Scientific Officer of the PRECISE Office, coordinating our national precision medicine efforts. So without further ado, Prof. Chambers.
Thanks. Hi, sorry, I hadn't taken myself off mute. So Kenneth, thank you very much for the warm introduction. Thank you to colleagues for inviting me to talk today. What I'm going to talk about is orthogonal to the excellent presentation Sebastian has given about infectious disease. What I'm going to talk about predominantly is chronic disease and how we're trying to unravel its etiology through studies of DNA methylation. The talk's got three parts. First of all, I'm going to talk about why we should want to study DNA methylation as a regulatory feature. The second part is going to be given by my colleague Lakshmi Lakshmanan, who's going to talk about the technical challenges encountered in handling genetic studies of DNA methylation. And the third component will be some insights into the kind of discoveries and that we can make through this kind of genetics of methylation studies. So, starting point. What are we trying to do? Well, so I've been introduced as the chief scientist for the precision medicine effort. So what we're trying to do is we're trying to understand better what drives disease in the population at an individual and population level. Try to understand the antecedents so we can do better about prediction through better biomarkers and prevention through better targeted treatments. So we start with the general complexity. Most chronic diseases are unfortunately don't have a single etiological basis. They're complex and multifactorial. They've got an important genetic basis that's estimated to account for about 30% typically of the etiological fraction, but also a more important environmental component, environmental and behavioral. Now, genetic association studies have led to tremendous successes and help us identify thousands of genetic variants that contribute to our risk of developing chronic disease. And the large-scale genome-wide association studies now identify genetic variants that jointly probably account for a large fraction of the heritability of many traits and many chronic diseases. So GWAS has been a tremendous success, and we've all seen lots of papers come out. But there's still a fundamental limitation, which is while we've identified a set of genetic variants that might be determinants of disease trajectories, actually, in the large part, we have very little understanding of how those genetic variants link to disease. And whilst a subset of the disease variants do cause protein coding changes, the vast majority of genetic variants that predict future disease actually sit in regulatory regions, and we have limited insights into how those genetic variants are driving regulatory changes. So that's challenge number one. Understanding how disease-causing genetic variants are influencing genome regulation. Then we come to the lifestyle and environmental factors. Now, there we've got two problems. The first problem is, well, actually, at one level, we don't necessarily know what it is we need to measure about people's lifestyle or environment. We don't know whether we're measuring it well. We don't know always what the exposures are that might be driving disease. So if you like, we need a biomarker that helps us get further upstream, closer to what might be the underlying causal exposure. And the second angle on lifestyle and environment is it's not simply the case that you get exposed to toxic atmospheric pollution, and that gives you suddenly lung cancer. There has to be a biological pathway that links that environmental exposure to the disease outcome. So again, we're looking for changes in genome regulation, genome biology that link the exposure to the disease endpoint. So there are several tracks for pursuing this, and one track that our group has been very interested in is using DNA methylation as a biomarker for genome regulation and hence underlying genetic exposures and environmental exposures. And for those who aren't so familiar with genome regulation, with DNA methylation, in brief, it effectively acts as light switches on the genome. Most cells in our body have fundamentally the same DNA sequence, but they do very different things. They behave dynamically in response to environmental challenges. And those differential gene sets of gene activation are in part determined through changes in genome regulation that are programmed by labile changes to DNA methylation that act as light switches on the genome. So DNA methylation is a very upstream determinant of genome biology. It sits right on top of the DNA sequence and determines how the genome gets transcribed, how it then gets translated into proteomic and metabolomic functions. And importantly, DNA methylation sits at acts as a singular common pathway that can both detect the effects of underlying genetic variation in a very proximal way as well as the effects of environmental and other behavioral adverse exposures. So it acts as a single point at which we can assess the panoply of genetic and environmental exposures driving human disease in an upstream proximal way. So DNA methylation, yes, it's influenced by underlying genetic variation, but it's also, as I've emphasized, determined by environmental and lifestyle exposures. We can demonstrate, multiple studies have demonstrated, that DNA methylation at a cellular or tissue-based level is associated with a range of lifestyle exposures, from fertilization, pregnancy, infancy, young adulthood, and age-related changes later in life. And our group has demonstrated some of these and the potential utility of these for risk stratification and disease prediction. Here's a study from some years back in which we demonstrate how DNA methylation, disturbances in DNA methylation, are powerful predictors of future risk of diabetes. For example, if you're in the top quartile for disturbed methylation, you're at a twofold higher risk of developing diabetes, additive and independent to traditional risk factors. So it's great. So DNA methylation captures what's going on in our life course and may also be a good prognostic marker, but we also want to know whether these relationships are causal or consequential. And the consequential aspect is important is because DNA methylation may not just be a biomarker for the underlying exposure, but if it's truly part of the causal chain, then it also becomes a target for treatment. And that potential has been demonstrated very elegantly in the cancer etiology space, in which the etiological role of disturbances of DNA methylation is well established, and there are several drugs that specifically target disturbances of DNA methylation as their primary mechanism of action. So we're interested in demonstrating. We're interested then in understanding how environment shapes DNA methylation, both as a biomarker, but also to understand which of those DNA methylation disturbances might be causal, to give us new insights into disease mechanism and etiology, and also as potential drug targets. So I have a twofold motivation for wanting to better understand DNA methylation. First, we have a bunch of genetic variants that predict future disease, and I'm very interested to understand what are the molecular pathways that link that genetic exposure to the disease outcome. Secondly, I have a bunch of environmental and behavioral markers that are linked to disease, and I want to understand the mechanisms by which those environmental exposures determine a link to the disease outcomes. And I also want to be able to test causality, and to test those causal relationships, I need strong genetic instruments to carry out causal inference and Mendelian randomization type tests, so I can understand which of the multiple associations you get in epidemiological soup might actually be causally relevant. So the data set we're going to talk about in terms of the demonstration of the methodology and to demonstrate the scale of the problem we're trying to grapple with, the data set we're going to illustrate is the SG10K data set, which is our phase 1 national precision medicine data set comprising data and samples from about 10,000 odd people. Those 10,000 people come from 5 discrete cohorts, and you can't simply we put them under the umbrella SG10K. In statistical terms, you can't simply treat them as a single entity. You've got to treat them as 5 separate cohorts and then meta-analyze data. Now, these 5 separate cohorts have been sequenced, have got their genotypes quantified by whole genome sequencing, combination of 15x and 30x that Nick, Bertin and colleagues have led on. Fantastic job. Those same samples have also had quantification of DNA methylation using what is the best in class at the moment, microarray, the Illumina methyl epic array, comprising 850,000 DNA methylation sites measured at single base pair resolution, with content that represented when it was designed best in class, based upon enrichment for a range of regulatory features. So the challenge we're trying to address then is I want to understand, for the twin purposes of unraveling genetic basis of disease and unraveling the environmental pathways to disease, I want to understand the genetic architecture of DNA methylation. And here I've got an N by M matrix. I've got 10 million SNPs determined by whole genome sequencing. I've got 850,000 CpGs determined by Illumina microarray. And each one of those has to be tested in a pairwise fashion, which represents 8.5 trillion statistical tests. Now, I can't just do it once, unfortunately. I've emphasized we've got the 5 sub-cohorts involved in the study, so I've got to do it 5 times and then meta-analyze to take account of those 5 individual cohorts. I've then got to do a variety of subgroup and interaction analyses, which mean that I can't just do it once in each cohort. Actually, I've got to do it 12 times in each cohort, so that we can identify not just the main effects, but also cell-specific effects. So that means I've got to do the 8.5 trillion tests about 60 times. So we end up with 510 trillion statistical tests, which I had to look up. It's half a quadrillion. It's an awful lot of compute, and it generates an awful lot of storage. So one run generates about 150 terabytes, and the whole exercise is going to generate about 900 terabytes. This is a tremendous challenge, and I'm going to hand over to Lakshmi, who's going to talk you through how we have approached the problem.
Thank you, Prof. Good afternoon, everyone. I will quickly walk you through the method that we use to scale up this for this size of analysis. As soon as we go more than 1 million association tests, we can't rely on the inbuilt regression methods. So we need some specialized packages which can handle large data sets like ours. So a quick survey really indicates that there are a few packages available, and then we choose our method of choice from this available packages. So without going into too much details, each method uses certain mechanism to improve the speed or the efficiency. So for example, matrix EQTL, as the name indicates, it speeds up the process by avoiding the solving individual equations one by one, but instead solving them as one big matrix operation. The tools that came after that, matrix EQTL, build up on that, and they add additional features such as permutations to do multiple testings. In these cases, they use some special statistical approaches to approximate the distribution instead of actually generating them, thus saving huge amounts of time. And the latest tools use GPU instead of CPU, adding more to the speed. Okay. So to choose among these tools, we use an empirical approach. We choose a very small old data set and use 3 different tools to analyze the same data set. So what we observe is that matrix EQTL is the one that gave us consistent results with the previous gold standard method. And for some reason, the TensorQTL, which we also tried, gave some huge variation in the results which we could not understand. So based on this small data set analysis, we choose to go with matrix EQTL tool. Our procedure is quite simple, but it's just that we need to repeat it millions and millions of times. So for a single phenotype, in this case is a single methylation marker, we have to do an association test with 8 million SNPs after our quality control, and we need to repeat this for 5 cohorts. And as Prof mentioned earlier, we have to use a meta-analysis to get a summary of the whole data set, right? So this is a procedure, but since matrix EQTL works based on the matrix operations, we cannot do phenotypes one by one. So in this case, we combine them into a bunch like 25. We optimized for this number, which I will explain in a few slides. So we use matrix EQTL to test the association for a set of phenotypes with the whole set of genotypes. And then we segregate the results by individual CpGs. Just as with our association testing problem, we also have the meta-analysis problem. We need a tool that can handle a large data set and do the meta-analysis in an efficient way. So we choose a tool called Metal, which is quite fast for our large data set. And it uses the inverse variance as a standard method for meta-analysis, right? So now even with this optimized tools, we realized that our processes are consuming very minimal amount of the CPU while we were running this. So then we realized that we could parallelize them between multiple virtual CPUs or multiple cores using some already available packages like MCL apply and parallel. So which gave us an improvement of about 4 to 5 times, depending on when we use 6 CPUs. So this helped us a lot to improve the speed for our analysis. So combining all the points that I mentioned earlier, so our pipeline for a single compute job for 25 methylation markers is that to do association testing for all the 5 cohorts and then do meta-analysis and then compress and save the results. And then we optimized it. We tried out different data sizes like 25 methylation markers, 10 markers, 50 markers. So we don't depending on the amount of files they generate and the extent of time they take, we chose 25 CPGs, which works optimally for us. So this one, our optimized pipeline for 25 phenotypes takes about 4.5 hours, which is quite consistent. There is no variability in this because we are not doing anything stochastic. So the timing is quite consistent, how many times you repeat it. So now to scale this up, we have about, in the EPIC array, we have about 850,000 methylation markers. So we decided to parallelize this process by breaking them, doing analysis for a small set of methylation markers in each node so that we could scale it up to the cluster. So the standard way to scale it up is to use a cluster and submit all our jobs. But this method didn't work out ideally for us because when we did scale up in the Google Cloud, there was an unexplainable amount of variability in time consumption between the nodes. And also, since we are recruiting a whole cluster, there is a good chance of idle nodes and we pay extra money unnecessarily. So we drop this option and then choose an option of just simply using thousands of independent virtual machines, which is more controllable and their time consumption is quite consistent and there is no loss of speed. And so we just made an automated job submission script to manage, because we cannot manually submit 30,000-40,000 jobs for a single model. So this job is quite simple. So we fix a number of nodes that we want to use, and then initially we submit jobs in all these nodes. Then periodically we keep monitoring whether any nodes have completed the job. Then we reuse the nodes again and again to complete the 850,000 CPGs. So this basically, these nodes do the job, save the file in Google storage, and then self-delete themselves. So we don't pay any extra money more than what is necessary. Right? So based on the time consumption for a single job, we could estimate how much time and how much resources we need to do the whole 850,000 methylation markers. So if we use 1,000 computers, it takes about a week or 7 days to complete 850,000 markers. Of course, when we use Google VM, some of the virtual machines fail during the run. So we need to pick them up again and rerun these jobs. So in total, it takes 8 days if we use 1,000 virtual machines. But if we can use 4,000 machines, we can complete it in 2.5 days or 3 days. So this calculation, even though it's a theoretical calculation, when we did our first scale up, we did see it's quite agreeable with our expected timeline. So we could do it in 1 week using on average of 1,000 machines. But our constraint is the size. Again, each round of analysis generates about 150 terabytes of result files. Needless to say, we haven't started analyzing the results. We have just generated the results. So we need to go through this 150 terabytes of files multiple times to generate our results. And this is just for a single model. Like Prof mentioned, we need to do this 10-15 times. So you can understand the scale of the work needed for this project.
Thanks. Okay, so it's a big job. but it's fantastic that cloud solutions have enabled us to leverage up to this. So we're now, through a fantastic partnership with Google, probably going to be able to access 5,000 virtual machines simultaneously, each with 8 processors. So we're effectively running 40,000 processors concurrently. And we estimate that we'll have the main job completed within the next 4 to 6 weeks. Okay, so what? What's it going to show? Is it going to be worthwhile? And to whet people's appetites, I'm going to show the results of what we've done from our preliminary work, the results of the preliminary analyses done using historic samples and a smaller microarray, the 450 microarray. So we just published this in January as a study done using just 3,500 data from samples from 3,500 people, half European, half South Asian, with genotypes measured by genotyping array and DNA methylation measured by 450K methylation array. So a substantially smaller data set with less coverage. And that smaller data set generated 11.2 million unique SNP CpG associations at a p-value of less than 10 to the minus 14. That very stringent p-value to allow for the extent to allow full correction for the burden of multiple testing. And this is one simple way of trying to illustrate the findings. It's a so-called chessboard plot. On the y-axis, we've got the SNPs in their genomic locations. On the x-axis, the CpGs in their genomic locations. And each dot represents a point where there is a significant association between a SNP and a CpG, giving it the pairwise genomic location. The blue and green diagonal is not auto-generated. That represents the so-called cis associations, where the SNP and the CpG are right next door to each other. And that represents the bulk of the associations, local genomic regulation. The more interesting ones are the ones in red. And I'd highlight, for example, this very nice column. There's a set of SNPs on chromosome 16 that are associated with DNA methylation across the genome. Associated in trans, a set of SNPs on chromosome 16 associated with the regulation of the genome on multiple chromosomes. And the data set, this small data set, identifies 1,800 of these so-called trans-acting genetic loci. And if we pile them up and say, well, which are the ones that have got most evidence for trans-acting genome regulation, we start to see some messages coming out. Most of them are trans-acting nuclear regulatory pathways. For example, NF-κB1, REST, SENP7, well known to be nuclear regulatory proteins. And that is further confirmed. Further evidence to support that these trans-acting genetic factors are identifying nuclear regulatory pathways is if we can show that there's close overlap between the remote regulatory sites highlighted by the CpGs and the known binding sites of the nuclear regulatory proteins, demonstrated by previous ChIP-seq and equivalent experiments done in multiple cell lines. What else can we show? We can show that these genetic effects on methylation are reproducible across cell types. That's shown on the right-hand side. So on the x-axis, we've got the effect shown in whole blood. On the y-axis, we've got the effect top, neutrophils, second row, monocytes, third row, isolated visceral adipocytes, and fourth row, isolated subcutaneous adipocytes. And even though the sample size for these individual isolated cell experiments is small, only 60 to 100 samples typically, we see that the vast majority of SNP methylation relationships replicate across cell lines. And that's very powerful, because that means that these SNP methylation signatures can be used to help explore not just blood biology, but potentially the biology of other tissues as well. On the left-hand side, we've got the the effect sizes identified for these SNP methylation associations, and it's presented as the variance explained. And the point I'm trying to highlight here is the strength of the association. So the average effect of SNP on methylation is 10% with an interquartile range of 4 to 11.5. These are tremendously strong effects, and this represents the advantage of going all the way upstream. In the context of our conventional clinical GWAS studies, we're lucky if we find a SNP that explains 0.1 to 0.2% of phenotypic variation. That would represent a strong effect. By going all the way upstream, we're getting much closer to the causal variation, and so we're getting much stronger effects. So this becomes in our view, a really strong way of getting upstream to understand the direct regulatory effects. We can also show that the transacting SNPs and CPGs are commonly in sites of intra and interchromosomal contact, as evidenced by an enrichment in topocologically associated domains and also in high C contact points. And and now we get into the real, the biological excitement. So what we do then is we say, well, are these SNPs that influence methylation enriched for association in biological pathways? And so we look to see whether these SNP sets are enriched for association with expression QTL signatures, either in cis or trans, or protein quantitative trait loci, or clinical quantitative traits. And in each of these plots, we've got the first column, the cis-acting MEQTLs, on the right-hand column, the trans-acting MEQTLs, that is SNPs and CPGs on different chromosomes. The shaded area represents the expectations under the null hypothesis based upon permutation testing of the background SNP CPG pairs, and the red arrow represents the observed proportion of MEQTLs that are associated with either the relevant signature, whether that's a transcriptional signature, a protein, or a clinical quantitative trait. What we see is that both in cis and trans, the SNPs that are influencing DNA methylation are highly enriched for being functionally relevant across a range of metrics. So what can we do with these data? This is quite a simplistic illustration to help give proof of concept. What we want to do is we want to integrate it we want to use it to unravel the pathway linking a SNP to its disease trait. In this instance, SNP RS730775 with its disease endpoint rheumatoid arthritis. So what we see is that SNP RS730775 is associated, turns out to be a transacting SNP and influences DNA methylation extensively in trans. And these brown boxes represent the transacting signature. We layer on additional molecular layers. We now incorporate eQTL data. And we can see that RS730775 is associated in cis with expression of a fairly well-known interacting protein, NF-kappa-B-IE. NF-kappa-B-IE shows covariation, interacts and shows covariation with NF-kappa-B1, which is well known as a key nuclear regulatory protein. And it turns out that these transacting methylation sites that are associated with RS730775 are strongly enriched for falling within the binding sites, the known binding sites of NF-kappa-B1. So that we're therefore walking from the SNP to its cis-EQTL to its primary interacting protein NF-kappa-B1 to its transacting genome regulation. And then in turn, we can see that there are a number of genes in the vicinity of these methylation marks in trans that are covarying with genetic variation at RS730775. So we're walking the full journey from cis regulation of gene expression through to trans regulation of gene expression through the interacting proteins and the respective DNA binding sites. And we can use genetic association, Mendelian randomization to demonstrate co-localization of those various molecular signatures. Now I showed quite a simple example to provide that sort of, yeah, that makes sense because it's building upon known biology. But of course, there are 1,847 of these transacting pathways. And at many of the genetic loci identified with transacting signatures, the local cis partner based upon a nucleotide signature has not been previously identified to be part of a nuclear regulatory pathway. So this highlights a treasure trove for biological discovery. And we've been able to demonstrate provide wet lab evidence to substantiate this proposition by doing targeted chip-seq experiments to validate some of the statistically inferred biological pathways. So I'm going to wrap up there and conclude. We believe that studies of DNA methylation are incredibly powerful as a key stepping stone linking genetic and environmental exposures to phenotypic traits. Studies of DNA methylation are non-trivial. The genome-wide evaluation of DNA methylation is like doing millions of genome-wide associations studies concurrently, and has tremendous computational demands, but these are now solvable with commercial cloud solutions. And the output of these kind of studies is a treasure trove of nuclear regulatory pathways that provide new insights into disease etiology and the opportunities to identify causal pathways underlying disease. So thank you for your attention.
Right. Thanks, John, and thanks, Lakshmi, for this really interesting talk. And quite a nice way to actually use DNA methylation sort of both as a proxy for the effect of the environment, but also as basically the mechanism by which the regulation operates. And then on that, one of the question that came out is, so you mentioned wanting to look at causality, and so have causal interference method like Mendelian randomization been used in epigenetic studies?
Oh yeah, they have, very much so. And you start with a simple Mendelian randomization to say, does the SNP influencing phenotype also influence the DNA methylation mark, and these other directions of effect approximately what you'd expect. But given LB between CPG sites and between genetic markers, what you then actually got to show is it's the same genetic variant that is explaining both the CPG and the phenotypic trait, that is so-called co-localization. And there are a range of tools now that enable that to be implemented at scale. So yeah, it's a well-established, but very dynamic and very exciting space.
Nice, thank you. All right, I think this is something you've sort of addressed with one of your analysis, but one of the questions that often come up is, so you'd expect methylation to vary between cell type, unlike the genome, which is shared across all the cell type. And so how do you sort of tackle, and how much is that an hindrance, like this expected variation across cell type? And
that's a great question. Again, I didn't go into the slides on that because I didn't want to introduce excess complexity. The studies done to date have all used pooled cell populations and have used pooled approaches to testing the association of genetic variation with DNA methylation. As a result, what comes out are effects that are common and shared across cell lines within that mixed cell population. So it's therefore perhaps not surprising that when you then say, okay, well, does this same effect also operate in an individual white cell subset like CD4 or CD8 or neutrophils or monocytes, then the answer is yes, because you've discovered it in a pooled whole blood fashion. But it's also perhaps less surprising that, not necessarily surprising, that it's also present in other non-blood cell types, because we're identifying, if you like, the key master regulatory genetic effects. So what's the next round of experimentation has got to deconvolute cell-specific effects. It's using statistical approaches to say, well, actually, what are the SNP CPG relationships in neutrophils specifically, as opposed to CD4 or CD8 cells? And that's where the complexity of our current computational challenges come from, in that we're we're using interaction terms to understand cell-specific effects. And in doing so, having to run the same models repeatedly, incorporating different interaction terms to deconvolute those cell-specific effects. And then we don't know what we're going to find. We don't know to what extent we're going to find SNP-CpG relationships that are indeed only seen in CD4, CD8, or other specific cell types, because this is an area of experimentation that has not been previously pursued. But we think it's going to be a real step forward for the field to be able to answer this question for the first time properly.
Nice, thanks. Actually, and then so related to like a question that is asked, and then also like sort of like rebounding on your side. So it seems that like what's mostly captured here is sort of elements that are fairly conserved, like methylation is fairly conserved across cell type. And so when you were showing this huge matrix of like the transacting SNP to CpG, one thing that came to my mind is like, how much does the map, that arrays of that, sort of correlate with the chromatin conformation derived map, which also, because they are so sampling like sort of somehow shallowly, or finding like what's conserved across cell type. Is there a good correlation between those like CpG SNP relationship and then chromatin interaction map at their resolution?
Yeah, it's a great question. It's a great question, and I'm not going to go as far as chromatin. The chromatin interaction map is on the radar, but I'm going to start with a simpler question first. I'm going to start with a simple question. Let's take a well-known nuclear regulatory protein like NF-kappa B. What transacting genomic positions is NF-kappa B regulating? At the moment, our insights to that are limited to those of pooled cell populations primarily, or cell lines. With this experiment, we're going to say, well, here's a genetic instrument, a SNP that changes NF-kappa B function. We can be able to map out, these are the transacting sites that NF-kappa B influences in a CD8 cell, as opposed to a CD4 cell, as opposed to a lymphocyte, in a whole human population. And then we're going to be able to layer on that to say, well, okay, so NF-kappa B is influencing different parts of the genome slightly in different cell subsets. How does that then interact with other underlying exposures, such as the presence of diabetes, age, ethnicity, and et cetera, et cetera, et cetera. So we'll be able to understand how these environmental exposures or other genetic exposures are influencing the impact of NF-kappa B in a cell-specific kind of way. And yeah, it's not just that sort of transacting NF-kappa B on genome, but also potentially how understanding how chromatin conformation might be, how chromatin conformation might change between cell lines, and how epidemiological exposures might be impacting on these chromosome interactions. So I think this can be an incredibly powerful tool for unraveling genome regulation in population-based terms.
Nice. I actually also a related question, like sort of, I think also like looking at it like more globally again. So one of the questions was like, did you notice any difference in the association between acting DNA methylation and then repressed DNA methylation mark?
Any difference? So the MEQTLs pile up in regions of active DNA regulation. So they particularly pile up in enhanced promoter and active DNA regulation sites. If you want to refine the question, I can try and refine beyond that, but that's probably about a sort of a high level kind of response that I can give at this stage.
Yeah. So there
is definitely a relationship. There is definitely a relationship between the distribution of SNP-CpG interactions and
histone modification conducive to DNA methylation repression and activation.
Yeah.
All right. And then there was one quick question. Actually, yeah, it was quite sad. So for like the slide were a little bit grainy at time, and so one of the people like following us virtually asked if the slide could eventually be shared. So I don't think there is any medium for that, but John, those slides are not sort of like made publicly available somewhere, right?
So, well, some of them are publicly available. Some of them are published. I don't mind sending out a redacted set.
Okay. So
yeah,
maybe what I'll do is I'll encourage people to drop you an email if they were so interested and want to follow up.
Yeah, that'd be great.
All right. Well, thank you very much. And with that, I'll let Kenneth introduce the next and last speaker of this afternoon session.
All right, great. Thanks very much for the great talk. Our third speaker, and certainly not the least, is to bring us back to one of the reasons why we actually look at this track, in which we want to improve healthcare. All right, no better person to talk about this than Prof. Daniel Ting. So he's from the Sing Health. He's an associate professor from the Singapore National Eye Centre. He wears many hats, and one of them actually, he heads the Center Program for AI in Sing Health. All right, so you can see from his talk, he's going to talk about AI in healthcare. He's been a thought leader in actually using AI technologies for patient care. So without further ado, Prof. Daniel.
Hi everyone. Thanks for joining us here. Great to be the last speaker. I'm just gonna actually share some of the considerations and some of the experience that we have learned over the past 6 to 8 years during my journey in AI and deep learning. When it first, I mean, it didn't really quite first hit the world, but it kind of hit the healthcare space back in 6 to 8 years ago. So I mean, I'm Daniel. I'm actually from the Sing Health, Singapore Health Service, and I am also a retina surgeon working at the Singapore National Eye Centre. So perhaps this is some of my financial disclosure that I would like to disclose. And we know for the past 2.5 years, the world is severely hit by COVID-19 pandemic crisis, and it's caused millions and millions of deaths, millions and millions of people losing their family, their friends. And in the healthcare space, this is something that what we have experienced over the last 2 to 3 years' time. So the waiting time in the hospital system has significantly increased. And because a lot of the healthcare workers were asked to actually go to the front line to fight the COVID-19. And not only that, I mean, a lot of the elective patients had to actually wait and to be reprioritized about their care within the hospital system because of the urgency of the treatment. And some of them unfortunately suffer from the delay of the treatment, and then that results in morbidity and some even mortality. And of course, I mean, the other thing that we know is that AI and digital technology has actually played a significant part in our life, not just in the medical field, but also in the healthcare space. And during that time when COVID-19 first hit, a few of us actually started to think, how could we leverage on our past experience in AI and deep learning to share some of the experience with the world and quickly gather some of the evidence and the data to share with the world to better prepare the countries to fight the COVID-19 pandemic crisis. So if you look at the author list here, together with Larry Kareen, who was a Vice Provost Chair for Duke University in US, Victor Zhao, who's currently the President of National Academy of Medicine in US, and of course Professor Tian Huang, who's actually also went over to Tsinghua to head the founding the medicine in Tsinghua. We actually have published this paper just in March and April, just 2 months into the crisis that's hit the world. So basically, this article was also cited by the US CDC as a reference piece to actually look at how digital technology could actually be divided into 4 different big parts, namely the IoT, Internet of Things, big data, AI, and blockchain for both COVID-19 positive patients and the non-COVID-19 positive patients. So basically, these are some of the examples that's already been published and been described, been used, been adopted in the different countries worldwide back then when we actually published this. So some of the very famous examples that everyone here would know is the Johns Hopkins COVID-19 dashboard that I think everyone actually will look at this on a daily basis, including myself when the crisis first hit. And of course, during that time, March, April, COVID-19 hasn't quite hit the world yet. So how do we use big data analytics to actually predict how the COVID-19 can spread worldwide is something that is key to a lot of the countries, especially for those who hasn't actually got affected by COVID-19. So of course, the deep learning for CT scans, for chest X-ray, to detect COVID-19 is something also very important for many the radiology groups to explore. So of course now we are at the 4th industrial revolution. Mankind has gone through 4 different industrial revolutions. When I give talks, I always tell the audience, this is my favorite slide because without these slides, without this industrial revolution, we can never be standing here talking and enjoying our life, having fun with our family and friends. So STEAM is something that it actually came through as a first to actually mechanize the human labor. Second is electricity, followed by computing, which is the Alan Turing's the era, and then comes the AI. And I wouldn't even be surprised or to say that we're currently at the 4.5 to even marching towards the 5th industrial revolution. And AI is actually nothing new. It was first coined back in 1950s, followed by machine learning term was coined back in 1980s by someone called Arthur Samuel, right? So at the time already he said this, the machine, the computer should have the ability to learn without being explicitly programmed. But what is lacking back then, right? That actually has resulted in deep learning that can actually come to fruition 20 to 30 years later. The lack of GPUs, the lack of supercomputers. Of course, that's why we are all here in the supercomputing Asia, right? So basically, the advent of GPUs has resulted the surge in the deep learning applications in the world that we are living today. So if you talk about deep learning, you need to know these 3 godfathers, right? Deep learning's godfather, jointly, they have won the Turing Award back in 2019, which is the Nobel Prize equivalent for computing. And I'm sure all the audience here are all from the computing backgrounds, and you will know what the Turing Award is. This is a reference paper that has actually really, this is like the classic papers that everyone should read if you want to know what deep learning is. It has been now cited more than 40,000 times, and it says that deep learning actually allows the data to be analyzed by multiple processing layers, and each layer would have their individual ways to do the abstractions of the features from either from the image, from the speech, or from the natural language processing. So three men here, right? Joshua Bengio, Yann LeCun, and Geoffrey Hinton. I mean, so these are the names that you have to know and has changed a lot of things we do in life. And if you break down the CNN into the different layers, you can be broadly divided into 3 different layers. It's quite simple, right? If you actually put it in a very simplistic manner, it's the input layer, which is the output layer, and the middle layer, which is a hidden layer. So if you look at the different, if you dive down into the different hidden layers, of course, then you start having the, the convolutional filters, fully connected pooling layers, and so on and so forth. And if you actually look at the evolutions of the CNN over the past 30 years, it goes all the way up to 1,000 layers from like 7 layers. And now we are talking about how do we actually make it more efficient to use less layer to produce the same and comparable outcome, so that we don't have to burn so much of the GPU, so that we can actually save the world, and so that we can actually achieve more sustainability, like moving forward with the increasing use of the GPUs. So all these things is very common these days. SpaceX, Tesla. Not sure. I always like to ask the audience, anyone actually knows what the name of the top right corner is? If you can see, it's a little robot called the Jibo. It's an MIT invention. It was it was actually awarded the Times Best Invention back in 2017. And of course, the facial recognition was also used to predict crimes. Whether that's ethical or not, that's also another big, the debates that, the world is talking about. And of course, AlphaGo Zero has actually shocked the world about 6 to 7 years ago when it first published, right? So basically, what has happened is it actually beaten the world best Go player 3-0 straight match, right? And this is AlphaGo. That's the first version. And of course, the second version came just 18 months later, called the AlphaGo Zero. So what technique does it use? It's so scary and it's so revolutionary. So basically, the AlphaGo Zero uses something called the deep reinforcement learning technique that can actually learn by being self-supervised, by not having any human intervention with the historic data. So this is actually, both papers are published in Nature, and how is it affecting what we are actually doing in the healthcare space right now, right? How is AlphaGo Zero technique can be used in the healthcare space to help us in some of the domains that there wasn't any technique for. So basically, AlphaGo team has spun off another team called the AlphaFold team to actually use the same technique to look at how we can actually get the AI to predict how the protein can fold and unfold. And why is it important to predict how the protein can fold and unfold? If you ask a human to actually manually calculate how the protein can fold and unfold, it takes us 13.8 billion years to do it. It's just impossible. But with deep learning, there is an answer to it. Why is this important to know how the proteomics of the human being? Is because if we can find out the answer to that, potentially we can halt how the disease can progress, or more excitingly, you can also potentially stop the disease from happening. So those are the things that is actually created a lot of excitement when this paper came out. AlphaFold first was described back in 3 to 4 years ago. AlphaFold 2 was actually just published last year. As you can look at the bar chart on the bottom right corner of the screen, you see that AlphaFold 2, the performance has actually increased by 30% from the first version. And this is if you actually hit more than 90%, as you can see, they are actually quite close to the high 80s. Potentially, we can start seeing a lot of the Nobel Prize winning drugs coming out into the space. So of course, Isomorphic Lab was also again spun off by Demis Hassabis late last year because of this. So if you look at if you do the horizon scanning in the medical space, in the AI used in healthcare, AI has been used across different medical specialty, including cardiology, how people actually look at ECGs, how to detect like the specific cardiology problems. And what about the respirology, looking at different chest imaging, the retina imaging, and of course the dermatology related lesions. And in eye, we started the journey relatively quite early, about 6 to 8 years ago. If you look at all the co-authors here, they were all from they are like the top guns in the Google, the AI space right now. I mean, Lily Peng's and Dale Webster's, and those are one of the first few who actually published all these papers 6 to 8 years ago. Gushan et al. 2016 JAMA papers, how the retina imaging is effective using deep learning to detect the patients who need referral for diabetic eye problems. And those are the things that we thought it would be good for us to also collectively as a group, as a consensus AI group in the world, to come up with some of the observation that we did during that time back in 6 years ago. So basically, we look at some of the very important technical and the clinical considerations when you need to build the AI system for the use in the real world setting, not just in the academic setting. You can see a lot of the papers that is coming out in the field for the past 5 to 8 years. And if you ask the same research group, are you using this in your clinic? More than 90% of them will tell you a lot of the challenges that has actually halted the translation of the AI algorithm from the research space to the clinical space. So there's something important, the valley of death that we always talk about. What are these valley of deaths? So basically, if you want AI to survive, you need to know the ecosystem very well. AI can never stand alone, and I always repeat this in multiple of my presentations. So if you want AI to speak, you need to connect. If you need connection, you need 5G, 4G, 6G's telecommunications network. If you want AI to get embedded in your clinical flow, you need to actually design the AI as if tomorrow, if I see any patients in my clinic, this AI needs to behave like what you are doing in the clinic. And of course, we look at scanning across the horizon, we also see a lot of the different technology that's coming out in the space. The emerging technologies such as the AR, VR, of course, the new term is called metaverse right now. And the home monitoring devices, as we actually pushing towards more self-monitoring and patient-centric care, this is some of the future of the healthcare system that we're marching towards. And of course, right now, COVID has catalyzed a lot of these digital transformation within the healthcare space. And those are the papers that we actually published to see how the different digital technologies could be interconnected with each other, talk to each other to actually get embedded within the real world setting. It's really important to actually make sure that the things you build is something that you're gonna prepare to use the next day. It's very, very important for everyone to actually know that. And I just wanted to talk about some of the principles about how we can actually build AI algorithms to be clinically relevant, to be clinically useful, to be used within the real world setting. So I summarize this in 5 rights. And it is not a rocket science to know what these rights are, but I just wanted to summarize it so that when audience actually leave the talk today, you could at least know what I feel about how those AI systems is being built. First, asking the right questions. Where do you want to use it? How do you see this AI system is analyzing your technology? Is it in the cloud? Is it in the retina cameras? Is it in a chest mammogram machine? Or is it in your computer, desktop, mobile base? This is what we call the intended use environment. Of course, that's just the hardware part. What about your patient's population? What is the disease subgroup? Is this for the general population screening, or is it for the tertiary or quaternary populations that you're aiming for? It's very different when you actually were to build the effective and the robust data sets to simulate that. So it's very important to first identify your intended use environment. Of course, the right data, the right technical partners, the right architect. So I always say the data and the technical partners are like a marriage, right? A man and a woman will always need to communicate. Otherwise, if there's a communication breakdown, that results in divorce, right? What about the right architect, right? So how, once you have the right couple, how do you actually create the ecosystem for them to actually move forward, right? So the architect of the environment, the ecosystem, the living environment is very important. And of course, the right enablers, right? So those are some of the things that I actually always share, the 5 rights. So the first right, the intended use environment. So when I get requests to say, hey, Dan, I have this idea. Can you help me to build AI algorithms for the patients, say in cardiology or dermatology? The first few questions that always come into my mind is, what is the market size, right? So first, how prevalent is the condition that you're talking about, right? So this is what we are talking about, market size, because when we don't have the market size, it's hard to scale such technology. When it's hard to scale such technology, when you bring the bill back to your chief financial officer, you always get a black face. They will say, what is my return of investment of? Of course, when in healthcare, it's not about dollars and cents every time we talk about, the medical breakthrough. But again, on the flip side, it's very important for us to start helping the chief financial officer to come up with the dollar and cents that makes sense to actually sustain such technology when we start embedding this into the clinical environment. So of course, the conditions is one thing. The other thing is the availability of the medical devices, right? So the condition can be very common, but the devices that actually needs to capture such condition can be very expensive and not available. So those are the things you have to think through, the boxes that you have to think through when you actually start looking at building some AI solution for a particular condition. And always think outside Singapore. So of course, this talk would not be relevant if I'm actually giving it in China because they have their population to sustain the deployment of the AI solution. But for those who are like us, like working in Singapore, building the technology from Singapore, always think the world is the home. Not just for 5 million population, either in SingHealth, NHG or NUHS, but think of the regional countries because, just right next to us, we have India, we have Indonesia, they're all the top 5 population worldwide. Of course, when you tuck into China, US, UK, Europe, we have a lot more population there. So the other thing is literature review. Don't be a frog jumping under the well, beating your chest to say that this is the world best solution when there's already 10 out there that's describing exactly what you want to do. So do the homework, go through the extensive literature review. When you say you're the world first, you better make sure it's correct. Because when a lot of the papers, I serve in a few editorial boards as an associate editor. A lot of the things that the authors always like to claim is this is world first, this is the world best, this is, So tone down the languages because there's a lot of times, even if you do extensive search, you may have also again missed 1 or 2 papers is really out there. So make sure that you've done extensive search, and when you write and when you report, make sure you tone down the excitement of you wanting to sell the technology. So this is something that I also share with the audience who wants to actually publish in the high impact digital journals. So diabetes is one of the things that I just wanted to say. It's a big problem. By 2040, 1 in 10 people in the world would have diabetes. This is huge. So in this room, if I have 10 people, 1 out of 10 by 2040 would have diabetes. So this is a scary disease, right? And that is just one of the many, many examples that I wanted to highlight about the market size. Right data. When we want to use the data, how do you get access to the data? What data are you looking at? How are you going to de-identify, anonymize the data? And if you don't have enough data, how do you go about inviting your friends to jointly build an AI system together? If you have enough data to build an AI system, but you don't have enough data to test it, how do you then use other types of technical methodology to test the generalizability of your AI system? It's all the different, very important AI domain that we have to actually make sure we are looking at. Data cleaning is another big topic, right? If we want to learn, we need to learn from the right dictionary. Garbage in, garbage out. This is something that everyone would be very well aware, right? How do we get the good label? So of course that would go back to the respective clinical domains, and the clinical experts would need to be the one give these answers. What is label? What is truth? What is the true truth? Different truth could mean different thing to different people in different countries. So how do we actually get consensus from all these experts groups? This is why in the clinical space, there's always the clinical experts groups, the consensus group, the consensus guidelines, and the international guidelines to govern all these consensus, right? And what about the data size, the data phenotype? When I want to build the AI system, how much do I have to use as a positive versus the negative? In other words, if I want to detect a lung cancer from the chest imaging, how much data do I need to use to build a robust AI algorithm to detect patients with lung cancer? And are we looking just lung cancer as a whole, or are we looking at a specific subtype of the lung cancer, such as the small cell lung cancer? So those are the intended use environment that I want to bring the audience back to my first point. It's very important to make it extremely specific what you are going to do with your AI system, right? So this is very important. Of course, numbers, I always get to ask the questions. What is the minimal sample size I need to build the AI system? I get it all the time. Hey, doctor, I have like 300 to 400 patients and, 5 years. What do you think? I say, start with what you have first, right? Be realistic. Of course, if you can start there, if surprisingly, because the unique features of such conditions, you may actually hit the jackpot. You never know. So I would say, of course, before that, do make sure that all the data you collected does simulate the real world environment. Don't pick and choose the data that goes into the AI algorithm, and it would actually not be generalizable to the real world setting. So those are the things that's very important. And breakdown of the patients, the eyes images are important. One patient, if you repeatedly take 10,000 images versus 10,000 patients with one image, which one would you go for, right? I would go for the latter, of course, right? You have 10,000, we talk about phenotype, we talk about the diversity of the images, not one patient with the same 10,000 images. So that is not, even though you have big numbers, but it's not effective. What makes a good reference standard? When you want to build and when you want to look for good reference standards, that is the truth that I was referring to. What is the true truth and what is the good truth? This is some of the things that you should be considering. How is your data set being graded? How many, what's the numbers of the graders are grading that? And what's the experience of your graders grading it? Is it a medical student versus a resident specialist? Or is it a senior or junior professors, right? So those are the things that you have to actually take into consideration. And working with the right technical team and the technique. I have a few more slides here because this actually highlights a lot of the basic principle of how some AI system was designed in a very flawed fashion. If you actually first identify the technical team, these are some of the usual departments that you will knock on the doors, right? School of Computing, School of Engineering, the Infocom, and of course the industries and the emerging companies. And I've mentioned about the marriage between the men and the women. Same thing between the clinical world and the technical world. If you don't communicate enough, if you only see each other once every 6 months and once a year, this marriage is gone, right? Same thing. If you want to first establish a relationship between the AI team, the technical team, and the clinical team, if you don't even have once or twice a week conversation for the first 3 months, especially if you haven't even had any prior experience in the AI space, that's for the clinician. I can say that because I'm a clinician. This project will go down the drain because you will take a lot of time to just understand the technical terms, right? You also need to spend a lot of time to explain some of the medical jargons to your technical team. And vice versa, you also have to ask a lot of the technical jargons that you're not sure about and try to understand them. So those are the things that you have to actually speak very rigorously with your technical team to actually get it ironed out. And when we say the appropriate AI technique, so once we identify the right technical teams and the right clinical teams, then we have to start getting into the technical methodology. For most of the clinicians, you may not need to know this, but for the clinician who wants to do this, or for the technical AI scientist who wants to do AI in healthcare related projects, these are some of the things that is quite important, right? The data format, is it a structure versus unstructured? Is it imaging? Is it genomics? Is it multimodal? What machine learning techniques are we using? Or can we use deep learning? Can you use data for the deep learning training? Can you actually use the data and the image and the genetics to build a multi-model AI system? This is something that we constantly think about. But of course, if you were to translate such AI system into the real world practice, I would usually share the less is more. If you actually have 500 inputs to come up with an effective solution, when you want to deploy in your clinic the next day, you will never fly. When you go into the EMR system to see how the clinicians is entering into the electronic medical records, usually there's a lot of the data that is not being keyed in properly, right? Because of the busy schedule and not the routine schedule. So those are the things that if you have less input data, that's actually a better way to actually deploy. Make it simple. What about the statistical methods that we are talking about? US FDA has come up with this guideline, SAMD, back in 2018. If you were to actually submit any regulatory applications for your AI system, you need to show them what is the intended use environment. I've used this term 4 times today in this presentation because if you overlook the intended use environment, you can spend 3 or 4 years or 5 years of your effort and still not getting the right regulatory approval. Because when US FDA, when any regulatory body comes down to your AI solution, they will say, which types of cameras, which type of machine, what specific phase of the MRI, the contrast phase, or which specific type of the population you are referring to. So those are the very important things that you have to know. And many AI groups always like to only report the AUC, right? Area under the curve. So area under the curve, there's also a different types of AUC we are talking about. Are you AUROC? Is it AUPRC? And what about the sensitivity and specificity? You have to always lock your operating threshold using your training data sets and then test it on your testing data sets. Because reporting merely on the AUC for the testing data sets is not enough, because it doesn't tell us any clue when you deploy such system in the real world, how well or how bad the technology is going to perform. And of course, 95% confidence interval tell us how tight and how solid your results is. So this is something that is very important. And the last thing is generalizability that we always talk about. How generalizable is your AI solutions to other countries, to other devices, to other populations? So those are the different hot topics within the AI space we're talking about. What about when we build the AI system? You always see the terms training, validation, and testing. What is train, what is validate, and what is test? Of course, when the world first getting hit by the tsunami of AI papers, right? I would say that 2016 to 2019, you see an exponential increase in the different big journals, right? Lancet, Nature, Nature Medicine, Lancet Digital Health, Nature Biomedical Engineering. You see a lot of the papers coming out. But that time, different people will use different terminology to describe the same thing. Validation set versus the testing set, right? So right now, internationally speaking, I'm actually involved in a few consensus group we are trying to standardize the use of some of these nomenclatures. It's very important. If you train, validate, and test, you always see these being written in the paper: 60, 20, 20, 70, 20, 10, 80, 10, 10. And my take is, if today your AI system performs well in the independent testing data sets and it is generalizable to many, many different independent testing data sets, no matter whether you use 60, 20, 70, 20, 80, 10, it doesn't really matter. Because the analogy is that if your basketball player can play really well in your conference finals and win the MVP, it doesn't really matter how this basketball player was trained back in the college, whether it comes from the Duke College, the top colleges, or whether it comes from Kansas, for those who play basketball. Yeah. So those are the things that I just wanted to use this analogy to share, right? So the independent testing data sets and the generalizability for the AI performance, in my mind, is actually the key. Of course, so the right architect. So when I say, what is the right operational AI flow, right? Just imagine a patient stepping into the hospital system today. Where do you go first when you step in? Registration counter. Where do you go next? Oh, if you are seeing cardiology, perhaps you need to have a chest X-ray, ECG done, or blood pressure measure first. Then the third location will be seeing probably another imaging test before you see the cardiologist. And of course, different clinical specialty will have different flow. Same thing for AI system. Today, if you're going to analyze your chest X-ray, first, do you have to determine whether is it a front to the back shot or back to the front shot? That's what we call AP and PA shot, or even a lateral, which is coming from the side. So your AI system should actually be able to tell whether this is an AP, PA, or lateral. That's one. Your AI system should also be able to tell whether this image quality is of sufficient quality. This is what we call the diagnosability, or it's called the gradability. It's interchangeable, the terms. So if your chest X-ray is being done improperly, say some portable X-ray, patient cannot position themselves, half of the chest X-ray will be blurred. So is this of sufficient to diagnose something in the chest X-ray? The clear answer is no. So your AI system should also alert you that image quality is not sufficient, do not progress to the next stage to be analyzed. So this is the second big part about the AI system. When we have to build these features in. What about the pre-processing steps? So we also know when we want to actually pre-process the image. So when a chest X-ray comes in, sometimes we may even crop the ROI, the region of interest, before we analyze the image. Same thing for retina imaging. And do you need to color enhance the image to actually allow the AI system to better analyze the image? What types of CNNs do you want to use? Dense net, rest net, efficient net. There's so many nets out there. Which is the appropriate nets for appropriate tasks, right? What about the output? What is your intended use environment? What is your 1 and what is your 0? Very simple question. Define the 1 versus the 0. That's always what I told people to do in step 1. What is your 1 and what is your 0, right? So those are the things that is very important to make it very simplistic. But the simpler the question is, sometimes you find it is harder to answer, right? So this is some of the things that you have to run through your mind when you design this. Of course, when we want to explain the black box of AI, explainability is very important. How do you explain what the AI system say when the AI system say is a yes versus a no, right? How do you explain that? So of course, people start looking at visualization techniques, the heat maps. If you look at chest X-ray, some parts of the X-ray shows like there's a heat coming out. Oh yeah, perhaps that's the region where you have the lung tumor or lung cancer, right? So there's a different types of visualization techniques we talk about, but in my mind, the maturity of the visualization technique is not there yet. So I think there is still a lot of room to grow, right? And of course, I mentioned about setting the right operating threshold. that determines your success versus the failure of your technology. You can have an AUC of 98%, but if you set your operating threshold wrongly, it will never be deployed effectively in the real world setting. So you have to take into account how much false negative and false positive patients you can take for your individual healthcare setting. If you allow too much false positive to come in, you will drive up and you'll jack up your screening costs for your healthcare system. But if you allow too much false negative to be missed out, you'll cost a lot of the life or even the sight for ophthalmology, for instance. So those are the balance, and based on the clinically acceptable standards you have to actually determine. Of course, when you build algorithms, you also need to understand there's such thing called AI biases. How do you take into account all these biases when you build your AI algorithm? Male versus female, population, the different settings. So those are all the different potential biases that you can inject into your AI algorithms that you have to take into account for. So this is just one of the papers that I published back in 2017. It's in JAMA. So basically, this is a high-level architecture to actually illustrate how the AI operational system can look like. Image goes in, it goes through pre-processing steps, it goes into the CNN with multiple different layers or abstractions. You come out to the fully connected layers and the output layers, and of course, there's also explainability layers after that. So those are the different things. And of course, when you want to scale, when you want to clinically implement, there will be 3 different boxes that you have to talk about, right? One is the internal IT and cybersecurity ecosystem. What is safe? What is safe in terms of the IT environment? You have to discuss with your digital officer to actually co-design this. What about the regulatory? Anything that impacts patient safety needs to go through regulatory approval of the respective countries. Commercialization, that is how you generate your ROIs, return of investment, and the financial sustainability for your AI technology. And I just wanted to share that last year in SingHealth, we established the AI program with the vision to actually position ourselves as one of the global leaders in the AI innovation space in healthcare. Our vision is to actually really to enhance the patient's outcome and of course to make their journey more seamless so that patients can have increased satisfaction when they come to see us, right? So those are the things that we are looking at, the 4 big pillars, the R&D, the commercial, the integration, and to actually, of course, to nurture the next generation and to create a more AI and digitally literate environment and community. So this is something that, and we have also formed the AI Exco. So we have about 15 Exco members, 10 advisors, and 25 honorary AI committee members. So a total sample size of 50 from SingHealth, representing the different institutions. So SingHealth, as you know, is a big healthcare institution in Singapore. We actually have more than 30,000 staff that's working within SingHealth, and we catered 50 to 60% of the subsidized population in Singapore. So basically, those are the different things that we are doing, and we are doing not too bad, I must say, right? I mean, thanks to all my colleagues who's actually interested in this field. So far, we have actually secured more than 100 million of the research R&D grants. We published more than 50 papers across all 10 specialties in the high-impact journals, NEJM, JAMA, Lancet, Nature Biomedical Engineering, and we are also continuing to actually look at the newer emerging space, digital innovation space, right? Blockchain, NLPs. So globally speaking, this is a trend right now that's getting really hot in the AI deep learning space in health. Standardizing of the nomenclatures, there's a different types of consensus groups is happening now. Start AI, Consort AI and Spirit AI. It has actually, they published the papers, it's a joint publications in Nature Medicine, BMJ, and Lancet Digital Health, published 2 years ago, standardizing all the different prospective AI clinical trials you need to tick all the boxes that's published by the form. Start AI, Decide AI, Quadras AI are some of the consensus group that I'm personally involved in. The data privacy and the sharing, federated machine learning. If you don't have enough data, if Singapore only have 5 million population, how do we build AI models together with countries like US, China, UK, without them needing to send the data over? This is through the federated machine learning. And what about the blockchain, right? Metaverse blockchain is getting very, very hot. Cryptocurrency, right? Those are the different, the emerging areas is coming out. Cybersecurity. We talk about a lot of quantum computing today, right? I mean, a lot of the other speakers in other sessions talking about it. If we have a massive amount of processing power, right? How is that going to cause the threats in the cybersecurity space, right? How is the people with ill intention is going to use these supercomputers to do something bad? This is something that the whole world needs to be very careful and very cautious about, right? And the generative adversarial network, right? Sorry, not generative adversarial network, but adversarial network. How can I inject virus into the medical images? How can I inject virus to any pictures or any images that say for instance, my autonomous car needs to see? If I inject virus into the stop sign, can you imagine what's going to happen to my Tesla? If it's going to be in the driverless mode, it's going to crash. To the human eye, you may not even notice a single difference in terms of the pixels. But you can inject a lot of the noise, a lot of the viruses into the image and make the autonomous cars misperceive the sign. So this is something that we call adversarial network. Sorry about my typo. It's not GAN, but it's adversarial network. And adversarial attack, AA, right? And generalizability, I talked about it just now. Last mile implementation is really important to show clinical value. Explainable AI, again, medical, legal, and AI biases. Yeah, so, okay, so I mean, this is one of my last slides. So AI and blockchain is something that we are looking at, and blockchain use has been widely adopted in the healthcare space for the past 2 years because of COVID, vaccination, passport. So I just wanted to end my slides in this. We are currently in a very rapidly evolving and technology explosive era, right? This is a very exciting field that we should all be prepared to actually embrace, to continuously innovate and to look for new angle to build the AI system using all the very cool technology we have, quantum computers and GPUs. With this, I would like to thank the organizer for the kind invitation to speak. Thank you.
Thanks, Dan, for like really inspiring talk on like the promise of AI in healthcare. So, because we are probably a bit tight in time, I'll just bundle like 2 questions into one that sort of are related. One is really on like what's the current thinking of like the liability when a model is used, and also like sort of related to it is what kind of method, I think you've alluded to it, for coming to like an explanation of how an AI system come up with a diagnosis.
Yeah.
If you could like give us some thought on that briefly.
So for the last one, explainability AI, yeah. So I think the explainability is something that it can actually be divided into data-driven and image-driven. And how do you explain the results is usually something that we are looking at visualization techniques. Where is the heat map showing the positive lesions, and where is the data that actually shows is this with a higher predictive power to actually determine the outcome. So usually, say if we do the image-based analysis, we'll look at the heat map. But we look at the data model, then we'll start looking at the predictive power. So those are the explainability, the method that we use. So of course, when we start going into more technical stuff, so for the image-based explainability algorithms, we can talk about GrabCam. So this is something that is actually quite commonly used, the gradient class activation map. Occlusion test is one of the very older technique that we use. Soft attention map, the integrated gradient method, those are the more technical techniques that is commonly being used. Of course, if you look at some of the Google papers, they also look at ablation analysis. If you look at ablation analysis, meaning that if I show you a picture of chest X-ray, if I take away the heart, so basically they use something to block the heart, and then they test the AI algorithms again on the same image. If the image performance still remain the same, it shows that the AI system is perhaps not looking at the specific region when they're actually trying to analyze for the example. So this is what we call the ablation analysis that the Google teams likes to use a lot. Yeah, so I mean those are the different common visualization techniques that perhaps the medical, the people who are interested can try. Yeah. So of course the first one, I mean liability, I think is a big problem, right? So of course, if you look at liability, you also need to look at the intended use environment. So when we talk about AI system in general, it's broadly divided into 3 different big parts. One is fully autonomous, one is semi-autonomous, and one is clinical assistive and what we call clinical decision, the support system, CDS. So if you coming from the regulatory standpoint, of course, I would be extremely stringent when I want to approve something, it is fully autonomous, right? So because there is no more safeguard, no more gatekeeper after my AI system has analyzed the results. Of course, the semi-autonomous one, you can either want the AI system to look after the normals or the abnormals, depending how good or how bad your AI system is and what kind of intended use environment you're talking about. And of course, the clinical assistive tool is that this AI system is to only used to highlight some of the specific abnormal areas, but the last gatekeepers is always the human. So in other words, the humans still need to analyze 100% of the image, but before that, the image will really say, look at this area, it's actually abnormal, right? The heat map, the visualization technique will actually tell that, oh, please be careful. If you actually miss this, right? So please make sure you take another look in this region. So those are the 3 big parts. So that would actually determine how the liability would actually be taken into account. Yeah.
Right.
Yeah.
Thanks,
so those are the general nutshell. Yeah.
Thanks. Okay. So, well, it's been a really inspiring talk, and then I'll just quickly wrap up as the next session is to be starting soon, and then thanks our 5 speaker for providing so good insight into really the wide spectrum of how supercomputing infrastructure is being used and how it impacts sort of both biomedical research and practice. So from the fundamentals of gene regulation with John and Lakshmi, real-time acquisition of data and analysis to really ensure a worldwide fast response to the pandemic, and the late talk from Dan on really the inspiring prospect of using machine learning technique to augment the diagnosis in an healthcare setting. And with that, I'll thank everyone, and then see you around in the session later today.